Laboratory of Genetics and Intergroup Biology at the Salk Institute for Biological Studies. He's an associate professor. He received his bachelor's degree in computer science from the University of British Columbia. He then worked as a software developer at the Ensemble Genome Browser at the European Bioinformatics Institute that most of us have probably used. Professor McVicker did his PhD with Phil Green at the University of Washington, where he researched how selection shapes human genetic variation. He did his postdoc with John Pritchard at the University of Chicago and then Stanford when John moved. Professor McVicker studied how genetic variation affects gene regulation in chromatin in that position. In his current position in his laboratory, the long-term goal is to understand the regulatory function of non-coding human genetic variation. To that end, the McVicker laboratory uses a combination of naturally occurring genetic variation, genome perturbations, and computational methods to understand non-coding variation. Today, Graham is going to be talking to us about interrogating gene regulation with single-cell CRISPR perturbations. And with that, I'll hand it over to him.
Okay, thanks for the invitation to speak here. It's really nice to be able to speak to a local audience. And because it's a local audience, I thought I would switch things up a little bit and present on things I've never presented on before. So this is a couple of new research projects from my lab, and I'm really excited to share them with you today. I'm very happy to answer questions throughout, although I'm not going to be monitoring them myself on Zoom or at the end after the seminar. So as Mike mentioned, my laboratory uses multiple approaches to try to decipher non-coding human genetic variation and its effect on gene regulation. The three primary approaches that we use are to leverage naturally occurring genetic variation that's present in human populations. We use this variation to do things like QTL mapping of gene expression QTLs or chromatin accessibility QTLs. And we also leverage allele-specific chromatin accessibility and gene expression to try to understand which variants affect gene regulation and in which cells. We also, in the last several years, have turned to using genome perturbations, where we actually can perturb regions of the genome and then ask how these perturbations change gene expression. And finally, we've been relying on machine learning methods and statistical analysis to try to predict when a non-coding genetic variation will affect genome function and gene regulation. So in today's talk, I'm just going to focus on the center area of genome perturbations. And I'm going to talk about two projects in my lab in this area. So in the first project, I'm going to talk about how multiple enhancers work together to regulate gene expression. And in the second half of the talk, I'm going to talk about a new method from my lab called SuperbSeq, in which we can jointly profile CRISPR edits as well as the effects of these edits on transcriptomes. Okay, so for the first part, how do multiple enhancers work together to regulate gene expression? And the question really is, when you have a gene which is regulated by multiple distal cis regulatory elements. So here I'm just showing two cartoon boxes of cis regulatory elements that regulate a single gene. How do these enhancers work together to control the expression of the gene quantitatively? And this was a project which was led by two people in my lab. Jessica, who was a BISB PhD student who graduated just over a year ago. And she's now a postdoc in Peter Ku's lab at Cold Spring Harbor. And it was also led by an undergraduate in the lab, Karthik, who did his undergraduate degree in data science and biology here at UCSD and has now gone on to do a PhD at Sloan Kettering in New York. So both people left the West Coast a year ago and are now at the next stage in their careers in New York City. Okay, so when we looked into enhancer-enhancer interactions and how multiple enhancers control gene expression, there's actually a fair bit of recent literature on this topic. But honestly, if you dig into it, it's really confusing. They make different claims about the action of multiple enhancers on gene regulation. Some of them claim that enhancers act in a hierarchy. Others claim that enhancers act redundantly or synergistically, or that there's epistasis between enhancers. So there's a lot of jumbled terminology and a lot of imprecision in the existing papers. And I found that in general, they're inconclusive. And part of why they're inconclusive is almost all of the papers thus far have focused either on one or two different loci and the enhancers that regulate those genes. So I feel like the question hasn't been well posed or asked in general for many genes in the genome. So what we want to know is, do enhancers actually combine in a non-additive way? And like I mentioned prior, studies are inconclusive on this topic. So you can kind of conceive of three basic models of enhancer interaction, or I'm going to talk about three different models today. One is that enhancers combine in a log additive or a multiplicative way. So this might be your baseline gene expression. And when you have one enhancer, you have expression at this level here in yellow. If you just have the second enhancer, enhancer 2, you have expression in blue at this level. And when you have both of the enhancers active together, you can just sum their components in log space and end up with this combined expression when both enhancers are turned on. So I call that the log additive or multiplicative model. And in this case, we would say that there's no interactions between the enhancers. A different version of the model, which you might call synergistic enhancers, the combined effect of both of these enhancers is greater than just the sum of their two parts. And another version of this you might call redundant, is that when you add the second, the combined effect of both enhancers is no greater than just the individual effects of the two enhancers. And so you might imagine if you knocked out one enhancer, you wouldn't see any effect on gene expression because you still have the other one acting on it. And in combination, you don't get this additive increase. So one way we can now actually measure enhancer effects is we can perturb enhancers. And one way to perturb enhancers is with CRISPR-I. So CRISPR-I uses the CRISPR-Cas9 system, but instead of an enzymatic Cas9 enzyme that cleaves DNA, it's a disabled Cas9 enzyme, which just hones into the target region, but it doesn't actually result in any cleavage of DNA. And typically in CRISPR-I, this dCas9 is fused to a repressive domain such as CRAB, and you can get silencing of regulatory elements using CRISPR-I. So if we target CRISPR-I to a putative enhancer, we can ask whether this changes the expression of a target gene by measuring the expression of the gene after doing this perturbation. We can do things where we compare cells where we have a targeting guide RNA to cells which either lack a guide RNA or that have a non-targeting RNA, and ask if this perturbation changes the expression of the target gene. So there's a way to do this now that's quite high throughput, and it's a method which is frequently called perturb-seq, or sometimes called high throughput CRISPR regulatory screens. And the way this works is that we're not just introducing a single guide into cells, but we're typically introducing thousands of guides into cells at the same time using lentivirus, where each copy of the virus has a different guide present in it, and they infect the cells. And then after infecting the cells with these guides, and these cells are expressing dCas9 CRAB or another CRISPR-I effector, we can do single cell profiling of these cells to measure their transcriptomes. And at the same time we measure their transcriptomes, we're also measuring the guide RNA, which is present in these cells. And we can then try to associate the presence of the guide RNA with a change in expression at different genes in the genome. And potentially this technology can be used transcriptome-wide, although it has a reputation for generating quite noisy data. Okay, so in 2019, Molly Gasparini, who was in Jay Shinjuri's lab at the time, used this perturb-seq technology, or a variant of it, to probe enhancer candidate sequences and connect them to genes in the genome. So they designed guide RNAs to target putative enhancer sequences in the genome. And then they delivered them to K562 cells with lentivirus, and they delivered them at a high multiplicity of infection. And I'm going to come back to this point in a few slides. And then what they did is simply partition cells based on the presence or absence of a certain guide RNA of interest. So if they're looking at enhancer A, they could group all the cells where enhancer A was targeted and compare those cells to all the other cells where enhancer A was not targeted. And then you can analyze and say, does the presence of the guide targeting enhancer A have an effect on the expression of a gene of interest? And using this approach, they were able to connect a number of enhancers to the genes that they regulate in the genome. So just like when you have a single CRISPR perturbation, perturb-seq allows the effects of many. Yeah. Yeah, that's a awesome question. So in the paper, they actually do a power analysis and they show, if you have a fixed number of cells and a fixed budget, there's actually a huge benefit to having a high multiplicity of infection because you simply get way more cells that have a given guide present in them. So they actually did a multiplicity of infection of on average about 25. So on average, each cell gets about 25 different guides, although there's a lot of variance in that. And they do a power analysis to show that that is in fact beneficial, although there has been criticism of that approach and the fact that a high number of guides in a cell might result in other effects on the cell. So the cell might kind of panic and be like, hey, I have a lot of RNAs in this cell that are foreign, or something like that. But yeah, but it's a good point. Okay, so using Perturb-seq allows you basically to measure the effects of perturbing lots of enhancers on lots of genes. So that's pretty cool. You can partition cells by whether they have guide RNA A, targeting enhancer A, or guide RNA B, targeting enhancer B, or no guide RNA, and you can compare the resulting expression. Okay, so we developed a statistical model for using this kind of data for detecting enhancer effects on gene expression, and it's fairly similar to the model which was used in the Gasparini paper, I should state. So it's just a bit of a modification to the model that they used, but the general idea is that we use a generalized linear model to model transcription counts, so RNA-seq, single-cell RNA-seq counts, and we say that these single-cell RNA-seq counts are distributed as a negative binomial random variable with a mean mu and some dispersion. And so this is the linear portion of this generalized linear model. This is the linear predictor inside this exponential link function. And it has a number of terms in it. So this intercept you can kind of think of as like the baseline gene expression of the gene. This is the effect size of an enhancer. And this is the perturbation probability of that enhancer. And in most studies, they just set this to 1.0 if they see the presence of the guide in that cell. We also include scores for cell cycle because it turns out a major effect on gene expression in cycling cells like K562 cells is what stage of the cell cycle your cell happens to be in. And we can model this using tools that are based into Seurat to get cell cycle scores for S phase and G2 and M phases. We include those in the model to account for that. We also include the percentage of mitochondrial reads because this might indicate some technical issues or variability within cells. And then we have the scaling factor, which is an offset term, which just basically want to account for the number of UMIs that are sequenced per cell. So if one cell is sequenced at twice the depth, we want to take that into account. Okay, and then using this model, we can have a null hypothesis, and that's that the enhancer perturbation has no effect on gene expression. So that this term here, this enhancer effect size, is no different than 0. And under the alternative hypothesis, this enhancer effect size is significantly different from 0. Okay, and then something that I'm not including here for simplicity is that there's also a few other covariates that we include. Okay, so we can apply this to the Gasparini data, just looking one enhancer and one gene at a time. And these are what the results look like in the form of a quantile-quantile plot showing minus log 10 p-values. These are the expected p-values on the x-axis under the null hypothesis of no effect of any perturbation. And these are the p-values we actually observe. And these are the p-values they report on the Gasparini paper. So they're not so different than ours in blue. And the ones we observe are in red. And just as a nice control, we do things like we shuffle the guide identity. So we say like if we perturb the guides in the data, it looks like the null. That's a good sign. So that means like it's like a local guide effect. And then we can also look at if we just choose random genes from other chromosomes instead of the nearby gene, what do we see? And we see most of the time we don't see any effect. So it looks like the null. So this shows us that the model is basically working as a way to connect a single enhancer to a gene and measure the effect of that enhancer on gene expression. Okay, so a feature of the Gasparini paper that I mentioned earlier is that it has a high multiplicity of infection, or MOI. And the other nice feature of this data set is actually has a huge number of cells in it. So they have over 250,000 cells, which they used in this experiment. And they screened about almost 6,000 candidate enhancers in the experiment. And this just shows the number of guide RNAs per cell. So this is this MOI measure. Basically, it's near 25 on average, but there's a lot of variance. And then this is just the number of cells which have each perturbation here. So the reason why we thought these are nice features of the experiment is we realized it opens the possibility of measuring interactions between multiple enhancers. And the reason that it does this is because there's actually a substantial number of cells where if you're considering two different enhancers, both of those enhancers are perturbed within the same cell. And so this is a nice thing because now, instead of just looking at the effects of individual enhancers, we can look at the effects when the enhancers are jointly perturbed. So there are a group of cells which contain a perturbation not only to enhancer A, but also to enhancer B. So we modified the model to try to look at this, and it has a few additions to the prior model. So we still have this enhancer A perturbation probability or perturbation here term. We add a second term for a second enhancer, enhancer B. So these are the individual or marginal effects of these enhancers. And we can also look at the joint effect. So in the cells which have both perturbations, what is this effect size here? And the null hypothesis under this model, where we're looking specifically for an interaction term between enhancers, is this interaction term significantly different from 0? So in order to apply this model, we have to, though, define a set of pairs of enhancers where we think we actually have some reasonable power to be able to test them in the data set. Remember, the experiment wasn't actually designed for this purpose in mind. So we have to look, are there actually enough cells which have these joint perturbations present? So we define what we call the high confidence set, where both of the individual enhancers are nearby to a gene within a megabase, and they have a significant effect on their own on the expression of that gene. And from this, we got a total of 264 enhancer gene pairs. And then we try to look more broadly because we're also interested in enhancer redundancy. And if there's enhancer redundancy, we wouldn't necessarily see these in the individual perturbations, right? Because you need both enhancer perturbations, in that case, to see an effect on the gene. And so we look more broadly across a set where there's both enhancers within 1 megabase of a target gene and where the enhancers were jointly perturbed in a minimum of 10 cells in the data set as a bare minimum. And if we use these criteria, we have 46,000 pairs of enhancers and genes that we can test. Now we want to assess, using these criteria, do we actually have power to detect interactions? That's an important question, right? Because if we don't have any power at all, there's no point in doing the statistical test. So we did a set of simulations, actually a large number of simulations, which I'm going to gloss over today, where we try to simulate as accurately as possible what the data set actually looks like. And so we have a bunch of reference parameters that we can set. We can use this to generate effect sizes that are known in the simulation for enhancer A, enhancer B, and the interaction effect. And then, given these effect sizes and the baseline expression of the gene, we can simulate expression counts. So we get this expression count matrix, just like you would get from the real experiment. That we can then fit our model to this data and estimate the parameters and see how they, can we recapitulate the parameters that we put in. So just glossing over most of the detail here, it shows that we, in general, have decent power to detect interactions. So these are positive interaction effect sizes on the right and negative interaction effect sizes on the left. And these different curves correspond to different multiplicities of infection. And if we compare in our simulation, I would say that the 20 line probably looks most similar to the Gasparini experiment. So these would be if you had an experiment with even higher MOI, which for some guides, we do have higher MOIs. But, and then these are the enhancer interaction effect sizes. And these are sort of scaled, this range of minus 0.5 to minus 3 and plus 0.5 to 3 to be sort of similar to the effect sizes we observe for individual enhancers. And so for very weak effects down here, we have, I would say, modest power. So we are not actually very well equipped to detect weak interactions here. So we might detect, let's say, 10% of weak interactions with this strategy. But when we start having sort of moderate effect sizes, we think we have maybe 25, 30% power to detect them. So certainly this approach is going to miss a lot, but it should, given the number of things that we're testing, be sufficient power to detect some interactions if they do exist in the data set. Okay, so what does the results from applying this model to the real data actually look like? This is when we look at the high confidence set where we know the individual enhancers have an effect on gene expression. We want to know in combination, do they deviate from this additive expectation? And the answer is no. We don't see anything significant when we look at this high confidence set of enhancer pairs. So we don't see anything at all that is statistically significant. Now when we turn to the larger set, we see a few things across this large data set of 46,000 things that we tested that come out as being significant. So in total, 31 enhancer pairs show evidence for interactions in this large set here. That's a pretty small number. And we started digging into them individually and looking at them one by one to see what's going on here. And something we quickly noticed is that a lot of the significant interactions are driven by a single outlier cell. So this is a real example of a gene here, BABAM2. I'm not familiar with the gene before this, but this is in general what a lot of the results look like, where these are just, I'm just showing the cells which are jointly perturbed here for this gene. And they all look kind of like, you know, low expression. And then there's this one cell that just has crazy high expression. And so we think this is a problem with linear models in general, is that they're actually very prone to outliers. And so we can assess how prone they are to outliers using a measure called Cook's distance. The way Cook's distance works is you just individually omit each data point one at a time, and you ask, what effect did that data point have on your coefficient estimate? How big was the effect on your model? And you can see for most cells, well, omitting these guys did nothing, but omitting this one cell, well, this had a huge effect on our coefficient. And so that's problematic. This is, by this Cook's distance metric, this would be classified as an outlier. We decided to discard the signals which resulted from outliers using a pretty conservative criteria here, where we say, if the maximum Cook's distance for a cell is 5 times greater than the mean Cook's distance for the other cells, we're going to discard this data point. And when we do that, 20 of these 31 signals goes away. So most of them turn out to be driven by an outlier like this. There are 11 that remain after doing this outlier filtering. And so here's one of them, which looks interesting. So there's 2 enhancers for this gene TIM3. When you perturb them on their own, they don't seem to do anything. But when you perturb them together, you get a big reduction in the expression of TIM3 to the point where it doesn't look like it's even expressed anymore. And then here's an example of one of these. We have 10 positive interaction effects, and here's what they look like. I will say that they look weird to me. The case is generally that the gene has fairly low expression, and there's no effect when there's a change to a perturbation of enhancer 1, no effect when there's perturbation of enhancer 2. But when you perturb both of them, all of a sudden it has high expression. And this is what they all look like. And so I would say they don't look like enhancers to me, because it doesn't make sense that if you silence 2 enhancers, all of a sudden the expression of the gene goes up, right? That's more consistent with some kind of repression of gene expression or some kind of artifact in the data that we haven't identified yet. So I don't think these are consistent with our notion of what enhancer interactions should look like. So that leaves this one. And we've dug into the one in some more detail, and this is what it looks like. So here's the gene in the genome browser. It's right here. And then here where the 2 enhancers are, they're actually quite far apart. So they're almost a megabase apart from each other. And it turns out neither of them are marked by H3K27 acetylation. So actually they don't look like a classic enhancer. And they're in separate topological associating domains. So they don't look like enhancers. So I don't know exactly what's going on with this example, but I would put my money on this more like being an artifact, like some weird off-target effects, or they're individually affecting different genes, which then affect the gene regulatory network, which then affect that gene. So I don't think personally this is a convincing example of the kind of thing we were looking for to begin with. So we wanted to sort of look at this finding in other ways. And so we did a totally orthogonal analysis using the very popular Informer machine learning model, which is a very capable model of predicting gene expression and other epigenomic marks in the genome. And then what we see is that, so in this experiment was done by Shush, who's a PhD student in the Koo lab at Cold Spring Harbor. and Jessica enlisted her help to help out with this analysis. But the idea is that basically you take a 200 KB input sequence into Informer, and you can scramble the sequences of each individual enhancer that you're interested in looking at. So we looked at the same sets of enhancers that we looked at with our experimental data, and just scrambled one enhancer, the other enhancer, or both enhancers together. And then we could see, did the effects of the individual enhancer perturbations and the joint perturbations differ from what you would expect from an additive model? And these are what the results look like. So this is our expected change in expression given a sort of the null additive model. And this is what our observed change in expression actually looks like for the joint scrambling of the enhancers. And so, this isn't real experiments. This is like all in silico analysis. And there's, you can debate how much you should trust Informer's predictions in these cases. But we think these results are still consistent with the null model, with there not really being a lot of evidence here for interactions between enhancers. So just to summarize this part, we think that we can conclude from what we've done that strong interactions between enhancers are not common, at least. And if they do exist, they've got to be kind of infrequent. We weren't able to look at every possible combination of enhancer pairs. There's millions of those. Or if they are common, they must have really small effect sizes. And if they have really small effect sizes, maybe that doesn't matter that much. Or, we only looked in K562 cells. So that's kind of a lame cell line. It's a cancer cell line. It's like mostly triploid, sometimes tetraploid, sometimes diploid. It's a model. Maybe if you looked at primary cell types under specific conditions or developmental transitions, or, maybe you would see enhancer interactions being important there, or maybe there's just, certain classes of enhancers that do have important interactions and we weren't generally looking at those. So there's all sorts of caveats, but I think we can conclude, that you can make some conclusions. And here we just did like a rough analysis saying that this is from the high confidence set, but saying if you have some prior belief about how common you think going into this study interactions were, and then you update your belief based on what we observed, your belief in the number of enhancer interactions should be lower. So if you went in thinking, well, half of enhancer pairs are going to have interactions, well, now your new updated belief should be 10%. So So I think, we developed a nice framework for trying to look at these enhancer interactions between enhancers. And I think it's a proof of concept if people generate data sets which are specifically designed to look at these interactions. So you could imagine like dual guide delivery where you perturb two enhancers of a gene on purpose, and that would be a better design rather than the data set we repurposed. And it kind of motivates more need for future experiments of that type. So, but I think that our results so far don't really support that there's a lot of enhancer interactions going on.
For this cell line, do you know the distribution of the number of enhancers per gene that were targeted in that data set? So
Yeah.
is it the case that the genes that you're picking up pairs on actually have 5 enhancers that are active in the cell line?
Right. We did look at that, and I don't have the figure in the talk, but it was a pretty reasonable distribution where it wasn't the case that we're like only looking at 10 genes or something. We're looking at a broad array of genes, and most of the genes have like 2, 3, 4 enhancers perturbed. But yeah, that was a reviewer question. So
Wasn't me.
Yeah. Okay.
So find what are some of them enough with deletion and actually validate the qualitative and at least have a system. But there's a lot of people who would love to have one locus they could study. I mean, some people make an entire career out of it.
Yeah. So just to repeat the question for Zoom people, for the ones that we did observe, where there was kind of that funny effect where perturbing both of the enhancers increased gene expression, would they be worth following up on experimentally? I think maybe. I feel skeptical personally about them. I don't believe them, but to really prove that, you would have to experimentally test them. And I think it could be worth doing. I would advocate doing more computational analysis of them first to see, is there some commonality of them that you could identify? But the problem we felt was that we didn't really have enough of them. There's like 10. to really do a computational analysis where we could pick up things like motifs or shared characteristics of them compared to other things. I think better data set first is what I would advocate for. Okay, so this paper is been posted on bioRxiv for about a year, and it's going to come out in Cell Genomics in about a week or two. But if you don't want to wait to see it there, you can check it out on bioRxiv. And I'm just starting the second part of the talk. How are we on time, Mike?
12:45.
Okay, good. So I think we're pretty good. The second half, I think, is shorter than the first half. So in the second half of the talk, I'm going to talk about another single cell perturbation project in the lab. And this time, we're actually doing the perturbations ourselves rather than doing computational analysis of perturbations that other people did. So in this project of joint profiling both CRISPR edits and transcriptomes, this project was led by Mickey, who's a BMS PhD student who's here in the audience today, and Brad, who's a postdoc in the lab, also here in the audience. And so I said, if there's questions on this, I'm going to field them to them. They're kind enough to show up. But Mickey led all the experimental assay and developed the experimental methods here, and Brad has done most of the computational work in this project. Okay, so I think it's sort of well known that CRISPR is a great tool, but it does have some problems that are known about. And one of the main problems that's known about for therapeutic applications of CRISPR, as well as general questions you might want to ask with CRISPR, is that it does have a lot of off-target effects. It doesn't always just hit where you want it to hit in the genome, and it's difficult to predict sometimes where these off-target effects might be. And many of the off-target effects may not affect genes directly. When they hit a gene and you know about it, it might be sort of easy to predict what it might do, but a lot of them might hit random places in the genome and like non-coding regions. And then in that case, it's often very difficult to predict, does this matter or not? Is it going to have an effect downstream on gene expression? Other problems with CRISPR is that actually many guides in CRISPR, for reasons that are still poorly understood, have very poor efficiency. So they may exert their effects only in a small percentage of cells where the cell actually receives the guide. And these on-target effect efficiencies are also really hard to predict. These efficiency issues increases noise in experiments, especially in experiments like Perturb-seq. And another problem with Perturb-seq related to this is that it doesn't read out the edit itself. It reads out the presence of the guide. And so just knowing that you got the guide doesn't guarantee that you got the perturbation that you wanted. And so this can add, if the guide efficiency is variable or quite low for some guides, this can add a lot of noise to your system. So we wanted to solve both of these problems. And so we thought of merging two different techniques, essentially. So we already described Perturb-seq, where you have a guide RNA and you're measuring RNA, and you're inferring the presence of on-target edits just by the presence of the guide, and you don't know anything about off-targets, but you get to measure gene expression. So that's nice. And then there's GuideSeq, which is a bulk assay, which tries to measure on-target and off-target edits and their locations in the genome, but it doesn't give you any information about the function of the edits. And it's a bulk assay, so you might not see edits that are sort of rare in your bulk population. Okay, so Mickey had the idea of combining these two approaches into something he calls Superb-seq. It's a single cell assay that gives you readouts both of the edits and of the mRNA. And so you get direct information about the edits, and we also get to measure the transcriptome. Okay, so the basis for Superb-seq is a pretty simple three-step protocol. So the first step, and I'm going to go through each of these in order, is actually labeling the edits. And the trick behind labeling the edits is you introduce Cas9 RNPs into cells. So this is a ribonucleoprotein complex, which is the Cas9 complex with the guide. And it cleaves the target site. And then we have a donor DNA sequence, which gets inserted at the target site. And this donor DNA sequence is a reporter. It's a T7 promoter, which we can then use to read out where the edit occurred in the genome. The second part of this is in situ transcription of these barcodes. So this is what's actually going to be able to give us the readout of those sites where the edit occurred. And then the final step is doing combinatorial single cell RNA-seq and getting readout of both the transcriptome and of the T7 RNAs, and then doing an analysis of this data. Okay, so let's focus on this first part, the Cas9 edit labeling with T7 promoters. So Mickey tried a variety of methods for doing this labeling using homology-free edits. And he tried a whole bunch of different designs of the donor sequence, and he made different optimizations to the flanking sequence of the donor. He tried different phage promoters, including T7, variants of T7, another one called SP6. He tried varying the length of the donor sequence. He also tried introducing bidirectional promoters, and he tried introducing some end modifications, and he tried a whole variety of this and did an extensive optimization. And then we quantified how well each of these optimizations actually performs by introducing the donor sequence plus ribonucleoprotein complex into cells with electroporation, extracting the genomic DNA, and just sequencing at the target site. And then we use a method called tide decomposition to actually estimate the frequencies of the different events that we get at the target site. And this is what the results look like from his different designs that he tried. And so some of them don't work, but some of them work pretty well, where he gets pretty high efficiency knock-ins. And so we actually decided to go with this design 2 design, which looks like this. It has the T7 promoter, has a little barcode downstream of it, and it has some flanking sequences as well. And it also has some of these end modifications. So he tried using this system to target a whole bunch of different loci. So I'm showing a whole bunch of different guides he designed and tested this out and quantified with TIDE here. And this is showing the location of showing the events we get at each of the target sites and their length of these events that are inferred from TIDE. And so oftentimes we do get a short indel at the target site, but most of the time we actually get the insertion of the donor. And so this plus 34 is the expected size of the donor in site. And so it works pretty well at most of the loci that he tried. And then we can actually further quantify it by qPCR. And so this is the fraction of cells which we're estimating are actually getting the donor sequence at each of these loci that he has targeted here. And so, and it's in different cell types. So K562, which I've been talking about, GM12878, primary T cells, and JERCAT T cells. And so he's able to get reasonably efficient insertions of these donor sequences in a variety of different cell types and a variety of different targets. So that's good. Okay, so now that that's been optimized, the next thing to do is to try to actually transcribe these T7s. And we want to try and transcribe them not in vitro. So in vitro transcription is typically, what T7 is used as a workhorse for doing, where you would extract DNA from cells and then introduce the components for in vitro transcription. So the T7 polymerase and NTPs and cofactors, and you would get in vitro transcription. But we actually want to do in situ transcription in fixed cells. And to do this, Mickey found a paper where they introduced what they call the zombie protocol, where you just permeabilize the cells and do PFA fixation. And then you add these components to this directly, and you actually get in situ transcription off of the T7 promoters that are present in the genome. And the reason we want this is because we wanted to retain the transcriptomes with the T7s that we're transcribing off of. Okay, and so he did a whole optimization of this T7 in situ transcription method. And so and you can quantify how well it's working by just doing qPCR on fixed cells where we didn't provide the donor sequence and comparing that to ones where we actually provided the donor sequence bearing the T7 promoter. And he did some optimization, and one of the optimizations includes temperature, and it turns out you can get better results when you do it at a bit of a higher temperature, so at 40 degrees instead of 37, and that's shown here. So this is if you have RNPs, but you don't have the donor DNA, you don't get very much product. But when you add the donor, you get a fair amount of product. In fact, we think the amount of product you get is sort of equivalent to what you see from ribosomal RNAs. So it's actually a very, very high abundance transcript in the mixture. And this is just showing the results for two different replicates at two different loci. So the gene SMARCA4 is one we targeted in CHD4. And so we get very high abundance T7 RNAs at these target sites. And then we can actually sequence these sites too. And this is just reads. So if we don't have the polymerase, this is what it looks like. There's no reads here. If we if we just have the RNP and the T7, but no donor sequence, we still don't get any reads. But then when we have the donor sequence plus the RNP plus T7 polymerase, we get lots of reads from this target site. So this shows that the in situ transcription is generating lots of these reads, and they have this T7 barcode in there. And this is just out of another site in another cell line, showing that it works. Okay, so now that we have those two components, we can put things together a bit more. So we can do the edit labeling. And Mickey designed guides, multiple guides to target 4 different components of chromatin remodeler complexes. So ARID1A, SMARCA4, CHD3, CHD4, and then did the in situ transcription, and then generated a single cell RNA-seq library using combinatorial barcoding. And so we use the split-seq method from Parse Biosciences. And the way it works is that you separate cells into wells, you barcode them, you pool them, you separate them, you add barcodes, you pool them, you separate them, you add barcodes. And the idea is that the products from every cell get unique barcodes on them using this process, and then you can sequence them and then analyze the reads and separate things out into the cells from which they came from. And part of the reason we wanted to use this approach is because we didn't want to rely on 10x, which for multiple reasons, but one of the reasons is that 10x relies on using poly-DT primers. So they amplify off of poly-A tails, which are on mature mRNAs, but we weren't sure that the T7 in vitro transcribed RNAs would have polyadenylation. And so we want to capture the non-polyadenylated fraction of RNA as well. And so the nice thing about the Parse kit is that it has in addition to poly-DT primers, it has random hexamer priming. And so in theory, you get all of the RNA species that are present in the cell. Okay, and so we did this experiment with 10,000 cells. And the next step is to actually analyze the data. And so Brad did this analysis, which I'm just going to really briefly summarize. So he adapts the standard Parse pipeline called split pipe to get these reads out with labeling of what cell each read came from. And then he looks for reads which, when they map to the genome, they don't map perfectly. And so the end of the read does not map. And so we can separate out the reads that have basically clipping, soft clipping on the ends to identify these putative T7 reads. But it turns out you get soft clipping from normal reads too, because the reference genome doesn't exactly match the genome of the cell that you're using, right? And so you frequently end up with funny soft clipping in different regions of the genome. So the next thing to do is to actually look at the soft clipped reads and say, do they have this T7 barcode in them that we put into the sequence, yes or no? And we can separate out the reads that way to get putative T7 reads from that. And then we can estimate the sensitivity and the specificity of this. And it looks to be by comparing cells where we have the donor sequence to ones where we didn't. And it looks like we can get pretty high sensitivity and specificity for separating out T7 reads from non-T7 reads. Okay, and then the next thing to do once we have the T7 reads is actually call where the edits are happening. And so Brad has developed a protocol for doing that. Basically, you get a variety of reads present at each edit site that might vary by their location and clipping. And there's like slight changes in the different reads. They don't all look the same, their orientation. So Brad does basically a graph-based clustering of these and then nominates like the most common event as sort of the canonical edit site. And you can, basically it's a way of grouping reads and just saying, we think these came from the same edit location in the genome. And then once we have this, we can call allelic editing. And so we can actually look for variations in the edit to infer when there's more than one edit present in the cell. And we can do this because our little donor sequence inserts in a random orientation. And so some fraction of the time, we can clearly see that there's inserts in both chromosomes because we can see T7 reads going in both directions. In addition to that, there's slight variability into the inserts when they occur. There might be an addition or a subtraction of 5' or 3' nucleotide, and that can be used to separate out the cells into different allelic counts. So we can actually try to count at a given edit site how many alleles that we have present there. And then the final step here is there is a bit of a problem in that we often have reads downstream of the T7 insertion site, which are sort of fragmented. And so we don't get this barcoding here, but we can see that there's excess reads downstream of the insert site. and this can confound our downstream estimates of gene expression. And so our simple hack for dealing with this right now is we just exclude all of the reads for quantification of the transcript that are within 500 base pairs of any edit site that we identify. So the net result from doing all this analysis is you get two matrices that are kind of a nice output. The first is your standard single cell RNA-seq read counts. So this is like what you would use to estimate gene expression from single cell RNA-seq. But alongside of that, you get edits that are present in the genome and an allelic count for each edit. So these are what the edit sites actually look like that we identify. So we identify all 7 on-target edit sites. And just to highlight some of them here, we had 2 guides targeting ARID1A and different locations of ARID1A. And you can see we have a lot of cells that contain edits of ARID1A at these 2 locations. There's other on-target ones like CHD4. We see over 1,000 cells carrying edits to CHD4, which was one of the sites that we targeted. But we also see all this other stuff, right? So we see 36 sites that we think are bona fide off-target events from these guides. And by the way, we designed guides using like GuideScan2. So these are supposed to be good, high-quality guides with minimal off-targets. And yet we see very frequent off-target events associated with some of the guides. We can compare these off-target edits with off-target prediction tools. So Brad tried comparing to 3 different tools. So CAS, AuthFinder, eCRISP, and COSMED all purport to be able to predict what off-targets might occur from a given guide. They all overpredict. So they all predict a ton of off-targets, most of which we don't actually observe. And then of the ones that they do predict, turns out most of the ones we observed, they didn't predict at all. And so they don't really seem to work in our hands, at least. We can also then associate the presence of edits with changes in gene expression. So these are edits. We have situations where we have no copies of ARID1A in a cell, or no edits to ARID1A, and then we have these knock-ins of these donor sequences to ARID1A we can identify, and we see a significant difference in the expression of ARID1A between these 2 classes. And then we can look at another target, CHD3, where we can group them into 0 edits, 1 edit, 2 edits, and we can see an association there between the edit copy. We can also do the same thing for off-target edits, though. So when looking at one of the guides that we had for SMARCA4, it had some really common off-target events. The second most common one was actually near this gene. I'm not sure if it's within the gene or not. CDC27, which is a known cell cycle regulator, which has been implicated in some cancers. And this is a very common off-target. And you can see this is showing the homology between the guide sequence and that sequence there. And so it does have a high degree of homology, but with just one mismatch, which is just adjacent to what you would call the seed region of the guide. So it has, most of the off-targets have recognizable homology, although many of them have several mismatches to the guide, including in the seed region, which is supposed to be the most important region. And we can associate the presence of the CDC27 edit, which we observed in 388 cells, with the expression of CDC27. We can actually see it does appear to change the expression of CDC27. So this is like concerning, right? Because people are designing CRISPR guides for therapeutic reasons, and you can have tons of off-targets that affect the expression of other genes, including cell cycle regulators, which you probably don't really want to mess with. And so Brad did some further analysis of this, and he looked at the gene sets from MSigB, the molecular signature database. And then he looked at whether when you have an edit to CDC27, remember this wasn't a target, but we could read out when we have edits to it. Is there an association between genes that are associated with G2M? And we can see, yes, in general, there's an enrichment of downregulation of genes associated with G2M when we have targeting of CDC27 here. And so that I think is concerning, but also interesting. And we can do some other things, like we can look at targets of the BAF complex. So ARID1A and SMARCA4 are both components of the BAF complex, which is an important chromatin remodeling complex. And we can see that when we have targeting of ARID1A and SMARCA4, we have an enrichment of differentially downregulated genes, which are targets of that complex, as you might expect. Okay, so that's where we are at with Superb-Seq so far. We're excited about it and we think it's ready for further applications. So if people are interested in trying out Superb-Seq, you can get in touch with us and we're going to start exploring further and larger applications of it now that we are fairly confident that it works well. It has several advantages, I think, over Perturb-Seq and of Guide-Seq, the method for identifying guides off targets. It reads out the edits, not the guides, unlike current Perturb-Seq protocols. And this lets us identify off-target events even in single cells, which is pretty cool. And we can determine the effects of these off-target effects. So we can see if they're associated with gene expression changes in the cells. And another cool thing about the protocol is that we don't actually have to use lentivirus for delivering these guides and doing this protocol. So we did everything with Cas9 RNPs. And we think that will make this work better in cell types where lentivirus doesn't tend to work very well. So we think, for example, it will work well in primary T cells where lentivirus is often tricky to get to work well. And it just uses off-the-shelf kits and reagents. So if you have the protocol from Mickey, we hope that it should be straightforward for other labs to adopt. Okay, so I'm going to conclude there. And I just want to thank the people that did all of the work. These are the current members of the lab, or some of them recently departed, but approximately current list of lab members. And the people highlighted in blue all contributed substantially to the Superb-Seq project, which I just finished talking about, and it was led by Mickey and Brad. And then the Enhancer Interactions project was led by Jessica and Karthik, and then Aaron Shoei from my lab also contributed, and then a member of the Ku lab, Shush, also contributed to the informer analysis I talked about. Oh, and the work I talked about today was mostly funded by this NHGRI award to the lab. Okay, with that, I'd like to conclude, and I'm happy to take questions.
Yes.
Great talk. I have two questions about the off-targeting effect that you observed with Superb-Seq. The first one is, do you have additional piece of experimental evidence validating that the very frequent off-target effects can be confirmed, such as by, for example, even TA cloning of those target sites and do Sanger sequencing?
Yeah, that's a good question. As far as I know, we haven't done that experiment yet, where we selected one of the off targets and then confirmed its existence with like cloning and sequencing, for example. But that could be easily done, I think.
Thanks. My second question is, in the Superb-Seq, also regarding those off targets that were not predicted by the previous off-targeting predictors. I wonder whether in the Superb-Seq protocol or the experiment, you include a no Cas9 control, because you're delivering this to a cancer cell line that's constantly proliferating. There could be many many double-stranded DNA breaks going on, and the T7 oligo that was co-transfected into the cells could be randomly integrated to those double-stranded break sites that are not necessarily part of the.
I don't think that's the case, because we can see homology between all of the off targets in our guide. Even though we don't identify them using homology, when we go and look, we see the homology to the guide sequence there. So I don't think it's just random double-strand breaks. I think it's Cas9 mediated double-strand breaks. Another reason I think that the tools don't work well at predicting them is because we used a tool to design the guides that tries to predict off targets, and it doesn't predict off targets. And so I think the ones that are occurring are the maybe less easy ones to see by these tools. If that makes sense. It's because we used tools to try to make guides that didn't have many off targets, yet they sort of have non-predictable off targets.
Got it. Thanks.
Question in the back here.
Really nice work. This Superb-Seq is a really interesting tool. I'm wondering if you could be missing off-target effects that just aren't integrating your T7 donor.
Yeah, I think we miss edits in general, right? So I think we can be really confident when we have an edit. And then we also have a set of control cells where we know they don't have edits because we can label a set of cells in the experiment which have no edits at all. But then there's this case where there's going to be situations where the T7 doesn't integrate, or we don't get transcription off the T7 at a very high level for whatever reason, and that we just don't see it. And so I have this idea that you could do a downstream computational analysis where you sort of seed your analysis with the edits and non-edits that you know about, use that to define clusters, and then assign cluster membership to all of the cells in sort of an iterative way, but seeding from the edits that you know about. And I think that could potentially work. So, but for sure, there's tons of cells in there that have edits that we don't know about still.
Cool, thank you.
I had a kind of follow-up to that question. Do you think the cells in which you do measure edits have additional edits that you can't measure within the same cell?
Yeah, I think there are additional edits that we don't measure in the same cell.
Okay. More question?
The 10x the parse split-seq protocol. The reason that a lot of folks use the DT primer is to avoid high copy things like ribosomal RNA. How does the split-seq compare in terms of coverage of legitimate mRNAs?
I'm not sure. We haven't done a direct comparison to what you might see with a 10x run, but so I'm not totally sure. But yeah, it is a concern, right? That if you're getting non-polyadenylated stuff, there's a reason why people do a poly-A selection. So there's another caveat with 10x as well, and that we weren't sure, because we do this on fixed material, and so we weren't sure how well it was going to work, because I thought there was some reason 10x might not be compatible with that. But Mickey probably has some thoughts. Okay, so the 10x. Okay, so 10x has, since we talked to them, have started selling fixation kits, but at the time we started doing it, they advised against it. So
You fixed them.
yeah.
Any final questions from the audience or on Zoom? Is there anything on Zoom? Okay, let's give Graham another round of applause. Thanks everyone for coming.
Thank you.